Good afternoon, everybody. How are you today? Thank you for joining us for our second library lunch and learn. Last month or 2 months ago, I can't believe it was already 2 months ago. My goodness, was health and wellness, and we hope that you have all taken some of the great advice from our last panel and looked more into the books that we discussed last time, and the authors were fabulous. I felt better from that one. I felt like I could learn something new in my older ages. And I also felt like I wasn't completely on my own, that I forget my car keys everywhere I go. So we've learned something in the past episode, and we hope that you will learn a lot today as well. Miriam, didn't you feel like you learned a lot in the last program?
Yes, it was truly inspiring.
Yes, it's always nice for people to realize they're not the only one that has like something that they struggle with or whatever. So we all got to do some yoga. Where while there won't be yoga today, there will be a lot of science and a lot of learning. And we have so many incredible people for you to hear today. And we are so happy for you to be here. We're having a beautiful day in the Northeast. So what better way to celebrate a beautiful day by having our own virtual science fair. You will see our featured speakers on the screen in front of you. We always want to remind you that we have this recorded. You will be getting the archives. You will be able to go back and view any of the information that we go over, all the title lists, so you don't have to feel like you have to scribble down all the information as the event is going on. We will send you the the list of all the titles in the program and everything. So feel free to reach out with any questions. But other than that, all you have to do next for the next hour or so is enjoy yourselves and learn something new and celebrate the wonderful titles that we have to talk about today and the great featured speakers. My colleague Miriam, who I'm always so happy to work on these with, is going to introduce our first speakers for you.
Um, Hello, everyone. Welcome on behalf of Penguin Random House's library marketing team. We're so glad that you've joined us for the Library Luncheon Room program today. We're excited to host this virtual science fair, and we have many special guests who'll be covering a broad range of scientific themes from public health and botany to food science and engineering. And it is now my great pleasure to introduce our first panelists, two esteemed librarians from the network of the National Library of Medicine, who will be presenting on citizen science. Tess Wilson is the All of Us Community Engagement Coordinator for the Middle Atlantic region of the network of the National Library of Medicine, based at the University of Pittsburgh's Health Sciences Library System. Tess works closely with public libraries and community-based organizations to integrate health information outreach into their programming. She is also a privacy advocate with the Library Freedom Project and an urban eco steward for her local park. With Tess is Margie Sheppard, the Outreach and Technology Coordinator for the network of the National Library of Medicine's Mid-Continental region, based at the University of Kansas Medical Center. She has an undergraduate degree in nursing and works with libraries and community organizations interested in improving the health and well-being of their communities by connecting them with the NLM products and updating their technology needs. Margie is part of the NNLM's Crowdsourcing and Citizen Science Working Group. Welcome, Margie and Tess.
Appreciate it. So you can go ahead and go to our next slide. So, hello everybody, and thank you so much for inviting us to talk about one of our favorite subjects, citizen science. One of our colleagues, Kelsey Cowles, was not able to be with us today, but she also was instrumental in a lot of the content in this presentation. Next slide. Okay, so before we really dive into our presentation, I'd like to give you a short introduction to our organization. The National Institutes of Health is the nation's leading medical research agency, and many of you might be more familiar with one of its more famous agencies, the National Cancer Institute. It's one of the many, many institutes and centers at NIH. The National Library of Medicine is also an institute at NIH. It's the world's largest biomedical library, which maintains and makes available a vast print collection and produces electronic information resources like Medline Plus and PubMed. NNLM is the network of the National Library of Medicine and is an outreach program of NLM. There are 8 medical libraries located in different regions of the United States, and we all work together to support the mission of NNLM by offering training, funding, information, resources, and much more. Slide. So first, what is citizen science? According to the Environmental Protection Agency, citizen science uses the collective strength of communities and the public to identify research questions, collect and analyze data, interpret results, and make new discoveries and develop technologies and applications. So in other words, citizen science is when global citizens unite to charge forth toward discovery, which we think is pretty exciting. Citizen science projects are extremely diverse, so no matter your audience or your needs, there is likely a suitable project out there for you. Citizen science participants can mark observations using an app. They can identify animals online or even don waterproof boots and wade through the wetlands. Next slide. So we're going to give you a little brief history of citizen science. So while the term might be new to some, citizen science has actually existed for centuries. And throughout scientific history, researchers have leveraged the power of the people to make valuable breakthroughs. And so because of the hard work of a lot of amateur scientists and curious observers, longstanding projects like the Audubon Society's Christmas bird count have achieved remarkable milestones and contributed to real policy change. These types of scientific undertakings are really unique in a few important ways. So because the capacity is increased exponentially when the workload is shared between scientists and members of the public, the pace of data collection has the potential to increase significantly. So these projects are also create unique opportunities for hands-on education and engagement, which can result in lasting impact on participants. And participation in citizen science is a potential for individual community members to engage physically with their environment, potentially strengthening their connection with the local ecosystems and encouraging a more intimate relationship with the world around them. And so for those reasons and many more, citizen science projects have become increasingly popular avenue of engagement. So we have a few highlighted here on the slide. So the Big Bumblebee Discovery Project was a 2014 initiative in the UK, and it's a really great example of how citizen scientists can get involved in zoology. It aimed to engage children and their parents with STEM subjects by enabling them to take part by observing and recording bumblebees visiting lavender flowers. Citizen science has also helped amateur astronomers discover new worlds from the comfort of their computer screen. The Plant Hunters Project on Zooniverse trains volunteers how to find undiscovered planets by looking at how the brightness of a star changes over time by reviewing publicly available Kepler telescope data. And finally, inspired by Pokemon Go, the Stream Tracker Project is recruiting people to check on small streams in their area and report information such as where is the stream located, is there water flowing through it, is it dry, or is it frozen? Next slide. So NNLM supports citizen science because we really feel like it's a really great way to break down some of the barriers between researchers and the public, and it helps foster a much better understanding of science and data. Citizen science is also a really great way to build community and connect diverse groups with shared interests. So if you're starting to think about doing a citizen science program at your library, you might be hesitant, it might intimidate you a bit or make you hesitate if you don't have much scientific experience. So I want to share just a small sample of the free information resources that are available to help you. So Kids Environment, Kids Health provides background information and easy to understand definitions and links to more advanced information and that could be really helpful for young citizen scientists involved in environmental health projects. So if you're looking at a project that involves any type of health information, I want to recommend Medline Plus. It's the perfect place to start. So it includes health topics on important issues like asthma, cancer, diabetes, etc., with information that is appropriate for all age groups, and it's often offered in multiple languages. There are also videos, health quizzes, games, and much more. OpenEye is a really cool site. It's an open access biomedical search engine. So if you need a visual for a display or a presentation, this is a really great place to go. And one of the really cool things that's housed there is NNLM's History of Medicine collection. You can access that through OpenEye. And one of my favorites, the National Center of Complementary and Integrative Health. It's the lead government agency for scientific research on diverse medical and healthcare systems, practices, and products that aren't generally considered part of mainstream conventional medicine. So let's say you're participating in the flu tracking project, Flu Near You. You could find evidence-based information about various flu remedies on this website. And of course, it's not on the screen, but I feel like I have to mention PubMed. It's a really great source of medical literature, but also literature in life sciences and beyond. Next slide. So I'm going to just talk very briefly about SciStarter. SciStarter is an online community that's dedicated to citizen science experiences for project managers and participants. You can find over 3,000 projects and events that are indexed on SciStarter. NLM and NNLM have partnered with SciStarter to promote health-related science projects and offer virtual science programming. You can view these projects and related resources at scistarter.org/NLM. Pass it off to Tess.
Thank you, Margie. So speaking of SciStarter, our number one tip for libraries who want to offer a citizen science program, but maybe aren't sure what project or projects might be a good fit, we recommend they head to scistarter.org and use their project finder tool. And there's a screenshot of what that looks like on the slide here. So this is a searchable database that's full of thousands of active projects, and you can filter by topic, age group, or you can even look specifically for projects that can be done, for example, over a lunch break, at a sports stadium, and a lot more. So it's a really helpful tool to use. Next slide, please. And another resource to take advantage of is the digital and free Librarian's Guide to Citizen Science. So this is a product of SciStarter and Arizona State University's partnership. And it was designed with library staff in mind. It's full of replicable examples and reputable resources. It's really a perfect guide for beginners, but also a useful tool for those with more experience as well. So it helps library staff learn more about citizen science and how that might connect with libraries, discover some STEM-related issues or issues of concern that might relate to your community, and much more. And that's, again, available for free and digital. So it couldn't be a more perfect resource. Next slide, please. Another thing we recommend is involving local conservation organizations. So local watershed associations, nature conservancies, parks departments, even local individual researchers and tree tenders can be really invaluable resources for citizen scientists. Staff from those organizations might be willing to serve as guest experts on a webinar, or they might even have suggestions for participation in their own projects. And one great way to bring in other groups and utilize existing networks is to partner with a Scouts group near you. These are all great groups to work with, and programming and volunteerism are already built into their objectives, so it works nicely for both entities. And there are even quite a few badges that your citizen science project could help them earn. And furthermore, there's an existing relationship between SciStarter and the Scouts that make it really easy to pick projects that are suited towards this group's specific needs. You can check out scistarter.com/girlscouts/info to learn more about that particular partnership. Next slide, please. And if you're looking for other ways to engage your community virtually, participating in Citizen Science Month is a great way to get your library involved with citizen science and crowdsourcing projects. And it requires very little on your end. This is a great place to get started. There are prepared posts and language for you already, so you can simply copy, paste, and reshare from your own page. And the Librarian's Guide to Citizen Science that I mentioned earlier also includes tools and details for planning Citizen Science Month activities. Citizen Science Month is coming up next month, actually, so get ready to participate. There's going to be a lot of fun resources available. Next slide, please. Great. Finally, we wanted to leave you with a couple great citizen science apps to share with your communities. iNaturalist is a joint initiative of the California Academy of Sciences and the National Geographic Society. And it's a really fantastic way to dive into citizen science. It's maybe my favorite app to share with folks. All you have to do is download it to your device. You take pictures of critters and plants that you observe in your neighborhood, and then those images are verified by second and third parties, other citizen scientists. And your observation joins others in a large growing database. So it's a really fun tool for families, and it's also a good introduction to observation practices, to peer review processes, and to species identification. And plus, it encourages you to engage with all the nature that surrounds us every day. iNaturalist also makes an app called Seek, which helps you identify plants. I use it in my garden regularly, and it's a really helpful app, especially fun to use on walks through the woods. And one way to bring these apps to your library would be to maybe create a post on social media introducing it to your patrons. You could even encourage them to share their observations on your page or compete to find the most new species. There are a lot of ways to gamify and engage with these particular apps. Last slide. And that is all from us. This is our information. We are more than happy to chat about citizen science. Like Margie said, this is one of our favorite topics. We could talk for hours about it. So it's been really wonderful talking with you all. There's a link to some resources there, nnlm.gov/ZEF. That's a little short link for you, some resources that we mentioned. Thank you all so much.
Thank you, Tess and Margie. We just have one follow-up question, wondering how you both got involved in citizen science.
I'll go ahead and go first. So I lived in central Kansas for quite a while, and we are on the monarch butterfly migration track. So I got involved trying to introduce the non-invasive milkweed into my garden, so it could be a place for them to feed on their way. They're on their long trek to South America. And then also I just got done doing the backyard bird count, and that takes place in February. And I just really enjoy being part of helping research. So I've got a little bit involved with the Alzheimer's project that's also host on our site, and just enjoy kind of checking it out and seeing the things that I can do to make a difference. And one of the things is, if you think about one scientist trying to cull data to make something move along faster in scientific or health research, it's really impossible. When you get 100,000 or 200,000 enthusiasts, it just rapidly goes down the track. So what about you, Tess?
So actually, I am originally from Kansas. So my interest in citizen science also started in Kansas. When I was really young, I was able to go on a herpetological trip with my middle school science teacher. And it was really fun. We went to Western Kansas and just kind of stomped around looking for reptiles and amphibians. And I kind of kept that passion with me when I moved to Pittsburgh, Pennsylvania, which is where I live now. And the way that I got to know my new community was through volunteering with conservation organizations. And one of them, the 9 Mile Run Watershed Association, I became pretty heavily involved with. I do monthly stream monitoring with them. I actually just went out yesterday and we collected some samples. And have really enjoyed getting to know my local watershed, because it's really fun to interact and sort of watch the stream change throughout the seasons and understand how that sort of affects our little ecosystem in my neighborhood. And I was just accepted to the Pennsylvania Master Naturalist program. So I'm really excited to get even more involved. So I've really enjoyed being able to be a part of a lot of citizen science projects, and look forward to a lot more.
Thank you both so much. These are very valuable resources, and we appreciate you shining a light on the collaborative work of citizen science volunteers and practitioners. Tess Wilson, Margie Sheppard, and a shout out to Kelsey Cowles of the Network of the National Library of Medicine. Thank you all for this inspiring presentation, and thank you for joining us today.
Thank you guys so much. We are just going to do a little virtual room change here while we're getting ready for our next panel. So just give us 1 second. Sorry, it just takes a minute here. That was very interesting and not something that we normally talk about with our, you know, on our program. So that was really fun. All right, hold on. Let me find our new speakers and I will be right with you. Feel free to grab some water. All right. And also, just so you know, we are going to send out their presentation in our wrap-up report. I know that some of you had asked that in the Q&A. Stephen, make a panelist. All right, here we go. It's like a virtual room change. Remember when people used to actually have to walk off a stage? And all right. Whoops, let me just find. All right, there you go. Okay, so remember you can hide panelists that aren't speaking to make the view a little bit better. And I'm going to go in and clean up some of the background here. But I just wanted to, before we get started, I just wanted to thank our first set. And now I want to welcome Stephen, George, and Carl for our second segment of the program today. Thank you guys for being here so much. Stephen Morrow is the Vice President and Executive Director at Dutton, and he's going to be talking today to two of the authors he's working on this span, George Zaidan and Carl Zimmer, and their books are so fascinating. Stephen promised me to do a full introduction, a formal introduction of both of their backgrounds and all of their backgrounds during the presentation. So I'm not going to do that today. I'm just going to turn the floor right over to them. And thank you all for being here, guys, and have a great time talking.
Thank you, Kelly. Thanks very much.
Thanks.
I don't know how formal an introduction this is going to be. I do want to say that thank you to everyone who loves libraries and who made this this talk about science topics possible today. That presentation by Margie and Tess on citizen science was really so interesting and enjoyable. I really appreciate that. So I'm Stephen Morrow. I'm actually not the, I always want to be a director, but I'm just an executive editor at Dutton, and I work on science books. I really love editing books that show readers the scientific heart of things. It's a great way to spend a couple of decades. So MIT trained chemist, science video producer, George Zaidan, and New York Times science columnist, award-winning journalist Carl Zimmer, artfully present deeper understandings of the world around us, from the mundane to the philosophical. Published this week in hardcover, Life's Edge by Carl is about the many ways we have tried to define, uncover, and create life. And published in hardcover last year and out in paperback next month, Ingredients by George is about the chemistry of the stuff we put in and on ourselves. In case it's not clear on your screen, this here is a Cheeto, and this over here is a brain organoid. I'll get to that in due course. These books inspire discussions with tweens in my home, such endlessly fascinating questions as, is stinky cheese good for you? Is it alive? One of them is now pretty obsessively reading those ingredient lists printed on food packaging. So, George, why are there so many ingredients in our food we can't pronounce? What is that stuff?
So all of that unpronounceable stuff, it falls into a few categories. There are vitamins and minerals, which are added to try and prevent nutritional deficiency diseases. The classic example there is iodine added to salt to try and prevent goiter. There are flavorings, which are added to try and make food taste better. There are many different types of texture modifying additives. So for example, most peanut butters will contain some sort of emulsifier to prevent the peanut solids from separating from the peanut oil. And then there are preservatives, which help prevent foods from going bad. And that's just a few of the categories. You can see the full list if you Google FDA list of food additives. I'm not sure I'd recommend doing that. You could spend days. But the real question is, why is that stuff in our food? And the answer is, we all want foods that are healthy, delicious, convenient, and cheap. But getting all four of those things into the same food is impossible. If you want peanut butter that doesn't separate out into two layers, you can either buy the all-natural stuff, spend 45 minutes stirring it, and then put it in the fridge, which personally I think makes it taste less good, or you can buy a peanut butter with an emulsifier in it. If you want everything to be all natural, you're going to spend more time and more money preparing your food. It's a trade-off. We just have not invented the end-all be-all superfood yet.
Yeah. Does it make sense to try to eat things with ingredients that have relatively few syllables, fewer ingredients overall? What about that?
Yeah, I know a lot of people who swear by that advice. I don't. So first of all, your grandma probably ate a lot of stuff that was easy to pronounce and bad for you, like Spam or pound cake or snickerdoodles. And your life expectancy is probably higher than your grandmother's was. So I'm not sure where this idea of idolizing grandma's diet comes from. Her wisdom, fine, but her diet, maybe not so much. Secondly, and I apologize in advance for my chemistry rant that I'm about to deliver here, but this whole, if you can't pronounce it, don't eat it thing, it drives me nuts because a single chemical can have 40 different names, and they can range from very easy to pronounce to completely impossible to pronounce. So for example, if I give you something to eat that contains 1-alpha D-glucopyranosylbeta 2-beta D-fructofuranoside, you think I was trying to kill you, but that's actually just another name for sugar. So then the name of a chemical tells you how the FDA regulates ingredients labels. It does not tell you how safe or unsafe the chemical is, especially if you're eating it over long periods of time. That is a much trickier thing to do, and I go into all the details in my book.
So let's get personal here, do you think of yourself becoming chemistry after your death?
Yeah. Yes. Yeah, so much chemistry happens after you die. I mean, rigor mortis is the classic example. That's the stiffening of your muscles. And it happens when you run out of ATP. Those are the molecular batteries that power pretty much everything in your body. ATP is necessary for your muscles to release. So when you run out of it, your muscles get stuck in this sort of, tight form. We all have a couple pounds of bacteria in our guts. That's our microbiome. When you were alive, you and these bacteria had a truce. You gave them a warm, safe place to live. You gave them lots of food, and they in turn help digest your food and provide other benefits that we're just discovering. When you die, you're not feeding your microbiome anymore. So they turn on you and they eat you from the inside out. They'll break out of your intestine, they spread through your body, and they start digesting you. I hope no one's eating, by the way, so I apologize in advance. Doing that, produces a lot of gases. One of those is hydrogen sulfide. That can react with hemoglobin, which is the sort of bus that carries oxygen through your bloodstream. And it turns it green, and that's where you get the sort of classic death green pallor. I think the craziest thing that happens after your death sometimes is bacteria can get to your brain and start digesting your brain and producing gases there. And those gases can't escape because your skull is rigid. So they end up creating bubbles in your brain. And on autopsy, it can look like Swiss cheese. It's actually called the Swiss cheese effect. So I try not to think about that particular one too much.
Yeah, me neither. How much more expensive is it to eat natural food?
It's a good question. So there was a randomized controlled trial that was done at NIH Hospital, I think a couple of years ago, in which two groups of people were fed either a diet that had lots of highly processed foods or a diet that had pretty much no processed food. Now, the headline result of that study was unsurprising. If you give people more processed food, they eat more food. You give someone a Butterfinger, they're going to eat more calories than if you give them spinach, right? That's sort of intuitive. But NIH had to actually go out and buy all these foods, and it's government money, so they accounted for every penny. And the researchers calculated that the cost of 2,000 highly processed calories was about $15, and the cost of 2,000 unprocessed calories was about $22. Now, if you're feeding a family of 4, that's about $10,000 a year, and that's a huge difference that I at least did not appreciate before I read that paper.
You might say it's where nutritional epidemiology hits your wallet. As promised, science offers insights into the mundane, like your grocery bill, as well as more philosophical questions, like how do you know if you have discovered the truth or not? George just mentioned the experimental procedure known as a randomized controlled trial. But before you even get to doing one of those, you have to agree on some fundamental definitions that frame the question you're trying to answer. Carl has written about the surprisingly difficult problem of defining life. Its blurry edges are so fascinating. So Carl, what is a brain organoid? Is it alive? Could it kill you?
Yeah, thank you, Steve. It's great to be here. Yeah, so the thing on the cover of my book is what's known as a brain organoid, and I think it's alive, but it's a kind of life that's very unfamiliar and very strange to us, and that we're just starting to discover. Basically, the way that you make a brain organoid is you just can take a skin cell from your skin, throw it in a dish, give it some chemicals. The chemicals basically reprogram it to develop into a neuron. You just switch the genes around and now it's a neuron, and starts to divide like the neurons in a developing brain in a human embryo. And they will divide and create a cluster of hundreds of thousands, maybe millions of neurons, all interconnected. And they will form a structure that's a lot like the structure of our cortex, the layers where we do our most important kind of thinking. These things produce brainwaves. They'll be sitting in a dish for months and start developing brainwaves, and those brainwaves mature kind of like we see with human babies. They start to produce chemicals that are only found once babies are born in their brains. So what are these things? Well, if you keep feeding them, they're alive. But are they alive in the sense that we're alive? I mean, when we define our own life, we think of our consciousness. Brain death is the definition of death legally. So here we have these skin cells that have become little brains in effect, and we don't know what to call them. We don't know what to make of them. Philosophers are pulling their hair out about these things. They're really important for doing studies on diseases of the brain, like autism. But what happens if they start, learning, for example, when we're not looking at them? What then? What do we call them then? And that's just one of the mysteries and paradoxes that I explore in my book.
I sometimes think that Cowles is one of the most tremendous resources for would-be science fiction novelists. So, libraries of the world take note. But speaking of, smart little things, how smart are slime molds? I know you wrote about them in your book.
Yeah, so people might be saying, I'm sorry, what are slime molds? So just to orient you, if you're walking out in the woods on a summer day and you look down and there's something weird on the ground, maybe it looks like dog's vomit, that's a slime mold. There are literally species of slime molds called dog's vomit. And there are many hundreds, maybe thousands of species of slime molds around the world, and each one is just a single cell, but it's a cell that forms a network of tentacles that can spread out, as big as a placemat. And they crawl around on the forest floor, feeding on the bacteria that they encounter, either on rotting logs or on the soil. And scientists love slime molds. They will take them and bring them into their labs and study them. You can just put them on a piece of paper and dry them out and store them away for months, and then add some water to them and they start to grow. You can put them in a petri dish and you can feed them. They really like oatmeal. So you put a little lump of oatmeal on a petri dish. They go and they feed on the bacteria that grows on the oatmeal. That's just a feature of your breakfast, just so you know. Anyway, these slime molds, again, it's just a single cell that's sort of pulsing around very slowly on the petri dish, has no brain, but it can solve problems. It can make decisions. So for example, scientists can create kind of a maze on a petri dish with various corridors that these slime molds have to go through. And they'll put the slime mold on one side and they'll put a little piece of oatmeal on the other. The slime mold will find a way through the maze. It will find eventually the shortest route through the maze to get to that oatmeal. So it shows you how even something that seems like an incredibly primitive form of life has this fundamental intelligence. So I would actually argue that there are some hallmarks of life, and one of them is this kind of basic intelligence, how to figure out your world and how to make decisions about what you're going to do next. That's the secret to staying alive.
There's been so much excitement in this household about the recent news from Mars, about that the Perseverance inhabitant of Mars now. What's the simplest thing that the Perseverance rover or its pet helicopter could find on Mars that would strongly suggest to you that there has been life on that planet, that life was still there?
One of the things that they're going to be looking for are funny looking rocks. They're going to be looking for rocks that look in cross section like a layer cake, layer after layer after layer stacked on top of each other. That's the kind of thing that bacteria do on Earth. In fact, the oldest fossils that we have of life on Earth, that are over 3.5 billion years old, those are these bacterial mats. So if you find those on Mars, you might say, okay, maybe there were bacteria on Mars like there were on the early Earth. Who knows? Maybe life started on Mars and then colonized Earth on a meteorite or something like that. There are other possible clues, but sometimes it's hard to know quite what to make of them. For example, bacteria on Earth give off methane sometimes when they are feeding on other kinds of carbon. Scientists have found weird plumes of methane already coming off of the surface of Mars, and they're not really sure what's making them. It could be geology, or it could be something alive under the surface of Mars, but they don't know yet. So sometimes the simplest forms are actually the most ambiguous. It's possible, that a space probe might actually just find real bacteria just growing on Mars, maybe scrape down underneath the crust and find them lurking there. But, we'd really need to bring them back to Earth to take a closer look, because who knows, it's conceivable that if you find bacteria on Mars, you actually found the descendants of microbes that hitchhiked on space probes that we've sent in earlier years to Mars. We don't know really how much we have contaminated the planet already with our own life. So we're going to have to really be careful. But it's also possible that if there is life on Mars, it's unlike anything that we've encountered on Earth. So we may not even know it if we see it, which is why we need to get a better sense of what it actually means to be alive.
That's great. I have to say again, it's been such a pleasure working with both these authors on these terrific books. I recommend them highly. We kind of used up all our time here, but thank you to everyone for listening, and I really thank Kelly again for the opportunity.
Thank you all very much. That was very interesting and fascinating. I love the concept of talking about basic intelligence. That was not something that we're used to listening to, and that was really fun, and contaminating Mars. So thank you. And like we said, we will be sending all of this out, and there will be an archive. We have both of the authors' social media handles if you have more questions. I think I have a lot more questions. But yeah, thank you. That was really fascinating. And I do think between the peanut butter and the rigor mortis and the slime molds, we have, I think you're right. There is a really good science fiction novel in here somewhere. So I think maybe that'll be our next panel. Thank you guys very much. Good luck with your books. We really loved hearing about them, and we really appreciate you being with us today. Thank you.
Thank you.
Thanks for having us.
Thanks. All right, we are going to switch to our next program. Hold on one second. Just give me one sec, sorry. Okay. Miriam, why don't you start talking about what our next panel is going to be while I get all the back end going. But have a great day, Stephen, Carl, and George. Thank you.
Thank you. Now it's my privilege to introduce Dr. Suzanne Simard, author of Finding the Mother Tree, Discovering the Wisdom of the Forest. A pioneer on the frontier of plant communication and intelligence, Susan Simard has been compared to Rachel Carson, hailed as a scientist who conveys complex, technical ideas in a way that is dazzling and profound. Born in the Monashee Mountains of British Columbia, Dr. Simard was educated at the University of British Columbia and Oregon State University. She is professor of forest ecology in the University of British Columbia's Faculty of Forestry. In Finding the Mother Tree, Dr. Simard brings us into her world, brilliantly illuminating the fascinating and vital truths about trees, asserting that they're not simply the source of timber or pulp, but are a complicated, interdependent circle of life. Part environmental scientific work and part memoir, a Publishers Weekly starred review calls Finding the Mother Tree educational, groundbreaking, a work that entrances, and author Michael Pollan says Finding the Mother Tree promises to change our understanding about what is really going on in the forest and other pressing mysteries about the real world. Welcome, Suzanne, and congratulations on the publication of your book. First question that I would like to ask is what inspired you to write Finding the Mother Tree, and what is a mother tree?
Miriam? Oh, Suzanne, are you there?
I'm here. Can you hear me?
Okay, yep, just making sure. Okay.
Can you see me? Do I need to do anything to?
Just give us 1 second. I'm going to, you're here, but I just want to, we lost your video. So just before you answer, let us just get you back up here. 1 second. Oh, there you are. Hold on. Okay, you should be back. Sorry, Miriam.
No worries.
All right, Suzanne, you're coming. Maybe just make sure your video is on.
I think, oh, okay. Yep. Camera, I think it's on. It says start my video and it's not showing me. I can't see my face, but.
Yes, we can't either.
Kelly, is there something I should do?
You know what, Suzanne, give me 1 second. Let's just, sorry, everybody. Let's just try one thing. Oh,
Oh,
there
there
you
you
are. Great.
are. Welcome
Yay.
All right,
officially.
perfect. All right, now I
Thank
will be
you.
quiet and you guys can chat.
So Suzanne, welcome again and congratulations. We wanted to ask you a very basic question of what inspired you to write Finding the Mother Tree, and also what is a mother tree?
Thanks, Miriam. It's so great to be here. And before I answer the question, I just want to thank everybody for inviting me, and just a shout out to librarians. And I have to say that when I was a kid, I was a super shy kid and kind of an outcast. And I hate to admit it, but the library was always a place I could go to feel at home and welcome. And it was quiet. I could hide in the stacks. I could read whatever I wanted. And the only other place that made me feel that comfortable was the forest, which of course I grew up in. And that's what I write about in my book. And so to answer your question, Miriam, what inspired me to write this book? I grew up in the forest, like I said. My grandfather was a horse logger. I spent all my time in the bush with my brother and sister. And so I went on eventually to university and became a forester. And what I learned in that profession and what I went on to see in the profession or the industry of forestry was so different than what I grew up with. And I started doing research on how forests work and realized very quickly that our approach to managing forests, which is very an agricultural based approach these days, was not right, that it was creating all these, what I was seeing, problems in the forest. And so I wanted to do research on trying to understand what the differences between, the forests we were cultivating in plantations versus our old primary forests. And, as I did this research, decades of it, and seeing all these researchers around me doing amazing work and understanding forests, and yet it never really made it into practice. We're still doing the same things. We're still making the same mistakes. In fact, we're making the mistakes even at a bigger, extensive scale. Widespread clear cutting of our old growth forests, our primary old forests. And so I wrote this book to try and get that research, which has been here for a long time, into the public mind, so that the public can make these decisions and pressure their governments to make better decisions for the people and for the planet. We still have a long ways to go, and I hope that my book is one small step towards getting there. So then, what is a mother tree? So this gets into the nuts and bolts of my research. So I mentioned that, in modern forestry, we harvest or we clear cut old forests and we create plantations. And I just wanted to say, even in my home province of British Columbia in Western Canada, which is sort of a place of old forests, right? I grew up in these old forests, and now we're actually a province of clear cuts. And we only have about 3% of our old forests left, old growth forests. To me, this is heartbreaking and a travesty. And it's not that, forests do grow back. And we can also create old growth like structures in forests, but we have to be smart about it. We have to understand what those structures and processes are. So a mother tree then in these old forests are simply just the biggest, oldest trees in the forest. They're those trees when you're walking in your forest, your favorite forest, or down your favorite boulevard, or even in your backyard or your park. And of course, I studied in really, wild forests, but it doesn't really matter where you are. But they're just those trees that your eye is attracted to. They're the biggest, oldest trees in the forest. And so what makes them mother trees? Well, what I study is the microbiome of the tree. And, I thought it was really interesting listening to the talk about, about the human being is a microbiome too. Like we have 2 pounds of microbes in our guts. That's amazing. It's the same with forests and trees. So the trees go into partnerships with other microbiomes to basically make their them function. And one of those microbes is called a mycorrhizal fungus. And these are fungi that live on the roots of trees, and they go into a partnership, and it's a symbiosis. And they both mutually, they benefit from each other, where this fungus, it's called a mycorrhiza, grows through the soil and picks up nutrients and water from the soil and brings it back to the tree and trades it for photosynthate, or carbon, the food, the basic food of life that trees fix in their leaves. And fungi don't have leaves, they can't photosynthesize, so they need this carbon from the tree. And the tree benefits from the fungus because it doesn't have to grow big, heavy roots to get at these scarce nutrients and water in the soil. So they go in these partnerships with these cheaper, energetically cheaper organisms called mycorrhizal fungi. So what I did in discovering the mother tree was I mapped what these fungi do. I mapped them in the forest, and I discovered that all of the trees in a forest are basically linked together by these mycorrhizal fungi. There's much more science behind this, but just imagine, if you're looking down, you're flying over your favorite forest and looking down, and if you could roll back the forest floor and create a map of the fungi, these networks of fungi that grow through the soil, gathering up water and nutrients for the trees, they also link the trees together. And what we found is that the biggest, oldest trees, those great big ones that catch your eye, have got the biggest root systems and the most fine roots that associate with these fungi. And so they're the ones, they're kind of like the center of the network. And if you can imagine other networks in your life, like, for example, a transportation network or an airplane network, where you've got great big hubs, which are like Chicago and New York, and the great big international airports, and they're linked to these smaller regional airports. It's the same thing in a tree, that in a forest, that the biggest hubs or the biggest Chicago airports are those big old trees. And we were able to figure this out by mapping them, mapping the network below ground. The other thing we did is a bunch of experiments trying to figure out, well, what do these old trees do? And with respect to, nurturing the forest. And we planted all these seedlings around these old trees where they could be linked into this mycorrhizal network or not. And we found that seedlings that were linked into the network of the old trees grew better. They were nurtured by the old trees. And not only that, our research, we carried on, and we found out that these old trees could actually recognize which seedlings were from their own seed, which were their own kin. Kinship is something we normally ascribe to humans or animals, but we've discovered it in trees as well. And so because of these many experiments that came to these conclusions, we started calling these trees mother trees, because they were mothering the forest and they were the centers of the forest. So much like in our own societies, where mothers have such a central role in nurturing and bringing up families and, the glue that holds society together. It's the same thing in forests.
I thank you so much for that. One of the things that really resonated with me about your book is it's also a very personal story, and it speaks to your development as a scientist. And I'm wondering how your experience as a woman working in the logging industry helped to shape your future.
Yeah, that's such a great question. When I started in the logging industry, of course, I grew up as a child in a logging family. But when I came into the industry, it was such a different thing. It wasn't a place that welcomed me. It was dominated by men. I was one of the first girls to actually get a job in the forest industry. They were just starting to hire girls, actually. This was in the late 70s and early 1980s. And even in my faculty of forestry, they were just starting to accept girls into the faculty. If you went and said, oh, I want to become a forester, they would actually discourage you from coming into the faculty because it was a faculty of men. But we did make our way in, and we made our way into the jobs. And so I was one of the first ones to get a job there. And I realized, as I was looking at forest practices, that it was missing this perspective that I had from growing up, which was about connection in the forest. Like if you walk in an old forest, you can see the roots are twined over each other. There are all kinds of plants that are huddled together. They're, collaborating with each other. And yet, in the industry, it was all about competition, right? It was about managing plantations so that you got rid of the competition, so you could only have, those trees that you wanted that would produce the most profit. And I just thought this was wrong-headed. And it was also kind of how the industry worked, right? It was, this very narrow view of how forests worked, which was governed by competition, that, things like collaboration didn't have a place in it, get rid of those plants. And so I knew that kind of, although I didn't articulate it in my mind at the time in my early 20s, that female perspective of understanding forests was completely missing. And so, my work really about the mother tree is about the networking, the social life of forests, which until then it was all about the individual tree that would dominate the forest. Which is still a mother tree, but it was, that the trees were viewed as individuals that were separate, that they were self-made, that they hogged whatever resources they could get, and whoever dominated was the winner. And so this played out in the industry as well. And so yeah, so I felt like an odd person there. I had to fight my way in, and it's taken decades until finally, here I am. I'm 60 years old, 4 decades later of research, and now these ideas are starting to become more known. And yeah, so it was a struggle, but it shaped me, right? It made me a fighter. It made me ask those deeper questions. Maybe if I hadn't had to fight so hard, I wouldn't have been able to ask those deeper questions.
Suzanne, thank you so much for sharing this story. I do want to share one line that truly resonated with me from your book. The forest is wired for wisdom, sentience, and healing. So the title of the book is Finding the Mother Tree, Discovering the Wisdom of the Forest, and this will be published in May. Thank you, Dr. Suzanne Simard, for joining us today.
Thank you. Did you want me to talk about that or no?
I'm afraid we've run out of time. We'll be switching over, but we hope that folks will have an opportunity to check out this wonderful title.
Thank you very much, Miriam.
Now over to Kelly.
Thank you, everybody. Thank you so much, Suzanne. Like I said, we're going to switch over. That was very inspiring. I loved the idea of the tree being the mother, and we thank you again for joining us today. We're just going to do another little change here. Give me 1 second, and we will get onto our last segment of the day, which is going to be very fun and very inspiring as well. So this PowerPoint might take a minute because it's visually stunning. So give us a second here. And I have to find my panelists. Hold on a second. All right. Okay. You are. Hello. Okay. Hi, Adam. We will be. I'm finding Heather.
All right.
Let's see. There she is. Oh. Okay. Sorry, I feel like it's extra clunky today. And okay. So hi, Adam and Heather. Thank you. This is going to be our last panel of the day. And what better way to end the program than with talking about the Curie Society, about a covert young team of young women that are not only members of the Curie Society, which is an organization dedicated to women that work in STEM, but they are also on high stakes missions to save the world. Let me just formally introduce both of you, and then we will let you get on with your program. But this is going to be fun, and you know, I love how our science fairs come together with hearing so many things that we weren't expecting to hear, and we're happy that you have joined us today. And after many years as an executive in the entertainment industry, Heather Einhorn, who is one of the co-creators, made the leap to co-found Einhorn's Epic Productions so she could create stories showcasing heroic young women and women characters. Heather is a co-creator of the popular scripted podcast that some of you might follow, Daughters of DC and Lethal Lit, a Tig Torres mystery, which was listed as a New York Times great podcast and was the first young adult scripted podcast, which is so exciting. She lives in New York City with her husband and creative partner, Adam Staffaroni, and she'd be happy to eat cookies for every meal of the day, which I'm only reading that part because I thought it would be fun if Adam's bio then ended with Adam loves to bake cookies every day, but it doesn't.
We'll make that change.
Yeah. And Adam has been creating comics in one form or another for the past 15 years. He's a graduate of Dartmouth College and received an MFA as part of the first ever graduating class at the Center for Cartoon Studies, which I really wanted to point that out because with having children in college, I think that's great. I think if they told me they wanted to study, if they wanted to major in cartoons, I would maybe be a little hesitant, but now I feel better about it. Adam is also one of the co-creators of the podcast Daughters of DC and Lethal Lit. And we're so happy you guys are here today, and we're really looking forward to talking with you about the Curie Society. This is a really fun project from MIT Press. So thank you very much for being here today.
Awesome. Thank you so much. We're excited to be here.
Yes, and just as a reminder, just tell me when to go to the next slide, and I will.
Thanks.
Awesome. Thanks so much for having us. Our graphic novel is called The Curie Society. This is the first ever action adventure graphic novel from MIT Press. We're super thrilled to be partnering with them on this amazing and fun work. It's years in the making, so we're really thrilled to be here today. Next slide. Exciting. So you heard a little bit about Adam and I, but our company, Einhorn's Epic Productions, or EEP, just a little bit about that first. We founded it to launch the next generation of heroic female franchise IP. And sort of what does that mean? Between Adam and I, we've worked at every comics publisher, DC Comics, Boom, Lionforge, we've run the whole gamut. And coming out of that world, you know, it was just pretty clear to us that there needed to be more female heroic characters. Most of the female heroic characters that are in existence right now are actually derivative of male characters. So it's Superman and Supergirl, Batman and Batgirl. So it's time that women get their own characters that aren't just derivative, weaker versions of male characters. So that's what we founded this business to do. Next slide. Okay, so a bit about just sort of how we got to the Curie Society. So Adam and I went to a Comic-Con panel at San Diego Comic-Con a few years ago. The panel was called Representations of Female STEM Characters in Media, which is an awesome topic, and that's sort of what brings us all here today. But you know, the panel was super interesting. Comics people, scientists. It was just sort of this combo of different super smart people talking about how can we do better? How can we get more strong female characters that are more sort of representative of the types of jobs and careers that women in STEM should be pursuing? You know, and somebody asked a question at the end of the panel, and the question was sort of, what are some of the steps we can take? And one of the panelists said, oh, well, you know, it would be great to get a female character that's the head of a lab, like kind of like Q from James Bond. And then somebody else said, well, you know, it would be actually really super awesome to get two female scientists together, like in a concept, just like two would be like awesome. And so Adam and I left that panel and we were like, wow, like if the bar is just getting two female scientists together, then that bar is like really way too low and we can do so much better than that. And then upon leaving that panel, we did, you know, we sort of knew all of this, but it sort of really all came together. We started researching like female heroic characters, you know, that are sort of like having these STEM careers. And when we went back through the history, it became clear that most of the female STEM characters in comics, as you can see from this slide, are villains. So they're not even heroes. And also you can see 1946, we have Dr. Poison, toxicology, 1966, Poison Ivy, we have botany, and then Dr. Harley Quinzel, a psychiatrist. And not only are these sort of over-sexualized, right, but they're villains. And it's not completely universal, but we noticed that most of the male STEM characters, like Tony Stark or Reed Richards, were the heroes. So, this was like between that panel and just all the thinking about everything that was happening, we were like, oh my God, we can do a lot better. Next slide. And so, that's sort of the sort of process that led us to come up with this idea of the Curie Society, which is an ensemble piece of many, many women working together in covert operations to save the world using their various STEM skills. And I know it's crazy to think that this is like that this is where the bar is right now. It should be so much higher. But this is the first time that anything like this has happened. So we're really, really thrilled to bring you an ensemble piece of multiple women working together in a team environment. Next slide. So the basic premise of the book is in 1903, Mary Curie founded a clandestine society where brilliant women could pursue the furthest reaches of their intellect. The Curie Society exists today in secret chapters at dozens of universities worldwide. So, this is I just want to clarify, this is historical fiction. Mary Curie did not actually start a secret society. We have gotten that question.
We fooled a few people.
Yeah, we have.
There. Yeah.
We have. So just clarifying, this is fiction. But yeah, kicking it to Adam to tell you a little bit more about the concept.
Yeah, we thought it was important to really build the history of the Curie Society into the story to show this is targeted at a YA audience, at girls who are maybe studying STEM in high school and wondering, is a STEM major the right thing for me when I go to college? Is a STEM career the right thing for me? So we wanted to not only show them these characters in order to inspire them, but also show that this Curie Society has been around for years. It's not just, women in science is not a new thing. This is something that has always been existed, even if those women didn't get the publicity and the public accolades and weren't talked about in the newspaper as much as their male peers. Next slide. We have our heroes. They are our new first-year recruits into the Curie Society at our fictional university, Edmonds University. You have Simone, who is our young, enthusiastic biologist. She's a excited, proud to be a geek for her STEM subject. And she's also entering college at 15 years old. We did some research on prodigy characters, female prodigy characters in media, and found out that it's about it's about 95% male characters whenever there's a prodigy. So we thought it was important to include a younger than normally college-aged recruit amongst our group. Second is Maya. She's our worldly mathematician. She has high-powered executives for parents who have she's traveled all over the globe with, but they're also, of course, very demanding and have very high expectations for their daughter. And our third recruit is Taj. She is the street smart tech programmer out of the group, and she has maybe a little bit more edge than the other two. Over on the right, we have the group of mentors at the Curie Society. We interviewed a lot of different working female scientists when we were putting together this book, and we were really impressed by how important mentorship was to all the women we talked to. Some of their stories were amazing, and they could trace back to a point either when they were in high school or college, when they were about to stop pursuing their field, but one mentor stepped in and really made the difference, all the difference in where they are now. So we wanted to make sure to show the importance of mentorship and get those mentor characters into the book. And of course, you can't have an action adventure graphic novel without some villains to battle against. So these are our villains at the bottom, and we built in some past history between the villain characters and the mentor characters, who were all together in the Curie Society when they were younger, but due to a parting of ways on ethical debates about science, they've now gone in really different directions over the years. Part of what we thought was really important was including those scientific ethical debates in the book. We wanted the sci-fi components of the book to really be cutting edge and even projected in maybe 10, 20 years into the future. Where will this technology evolve in the coming decades? And tracing back the villain characters, we wanted to say, okay, they took an ethical compromise or went a different way on the ethical debate when they were younger and fell down a slippery slope to where they were now, but to really set up those debates to engage our readers on these interesting ethics that pop up as technology evolves. Because every time a brand new technology like CRISPR is in the book, when that's invented, a new ethical debate rises up around it, because it's something that kind of raises a new series of questions or changes some presumptions that people had before that technology came along. Next slide. And to get all that science into the book, we consulted with an amazing panel of real scientists. And we were basing a lot of what we showed in the book off real experiments that we read about, real stories from the women scientists that we talked to. There's a glossary in the back of the book that goes into more depth about the real science behind a lot of these concepts. So for the curious readers, there's additional reading at the back to continue to educate them on these topics. But everything is integrated into the book in a fun, engaging way. And a big part of what we wanted to do was make it fun, just fun to read, and show people, show our young readers how fun it is to engage with STEM concepts. Next slide. And these are just some of the scientists we talked to. All different fields, different backgrounds, different interests, and science communicators and journalists, as well as professors and working researchers. We had an amazing team at Massive Science that helped us assemble this panel. And we just had a wonderful time. We learned so much talking to all of them, and had quite a big challenge in front of us in figuring out with our amazing writer and amazing art team how to incorporate all of that information into the book. Next slide. And of course, ending on a quote from our inspiration, Marie Curie. Nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. We learned a lot of amazing things about Marie Curie herself in the research and putting this book together. And one of the big ones was that she hated publicity. So having a secret society in her name seemed very, very fitting to the true inspiration behind the Curie Society.
Thank you. That was fun. This is very inspiring. And so you're right, something brand new to share with a whole generation of young readers and middle grade and young adult audiences. So thank you. We're really excited for this one. we will be talking about. It's coming April 27th. As you all know, the information will be in the PowerPoint. But thank you very much for I love how you listed the scientists that you talked to, and very inspiring. And I love how you said Marie Curie hated publicity. So I love that you gave her a secret society. It's so cool. Maybe she really did have one and nobody knows.
It could be. It could be.
So thank you very much for being with us today, Adam and Heather. We wish you the best of luck with this book and for being the last panel in our virtual science fair. That was very fun.
Thank you. Thanks. Yeah.
Thanks. We will talk to you soon. Thank you. Just before we go, we just wanted to thank all of you for joining us. Like we said, for our virtual science fair today. We hope you all learned something, got some ideas for your collection development, for programming. We certainly learned a lot. I know that these are not authors I normally listen to talk, and so it's fascinating for all of us. And when you get the PowerPoint and when you get the wrap up, we will send you all of the information today. And also we have a list of further reading for each of these topics covered today, such as nature, environmental science, life science, psychology and mental health, and physics. And so this will give you a little bit more of a robust collection of books within these disciplines. And we thank you for joining us. We have a really fun program coming up in May, which is going to go like that. So we hope you will register for that in advance. And Miriam's going to tell you a little bit about that. And again, thanks all for joining us for our second episode.
Yes, thank you again to our guest authors and editors and creators. On May 13th, we'll have the third Library Lunch and Learn program, and it's going to be focusing on planning for Pride Month. We have confirmed that we have a special guest librarian from the New York Public Library, Jason Bauman, the Associate Director for Collection Development and the Coordinator of Humanities and the LGBT Collections at NYPL, as well as the editor of this book, The Stonewall Reader, will be joining us on the 13th. So we hope that you register for that. Thank you again, and a big thank you, library colleagues, for all that you do to support authors, books, and readers everywhere. We hope to see you soon. And as
always, let us know if you have any questions, but thank you for joining us and have a wonderful rest of the day. Thank you, Miriam. Bye.